Hello everybody, how are we doing today? Today we're going to continue on gene expression and central dogma theory. As we said before in the other class, um, DNA speaks in four language. DNA speaks with four letters in its language: A, T, C, and G. Our body does not actually read and understand A, T, C, G. It actually understands proteins, which are our body, and our body actually, proteins actually speak, as we see down here, amino acids. So if you don't have this written down, please write this down. So DNA speaks ATCG, protein speaks amino acids. So we need this junction. We need something, this Rosetta Stone, this translator in the middle. The translator is this RNA. It's called ribonucleic acid. It's similar to DNA, but it's single-stranded. And it speaks four letters for its language, and it's A, U, C, and G. And go from one process to another process. We actually go through a thing called transcription. So we go from DNA to RNA. It's called transcription, which means all we do is we take one thing and we write it down verbatimly, as in if you're Mr. McNeil's class and you're learning a new sentence, you would write down the sentence in Spanish and then convert it later. It's not like you would see, no hablo español, and you would translate that right to English. You would actually just write it down and then translate it. So transcription just means to write it down. So we wrote down DNA, we converted it into a single RNA, which we'll talk about in a minute, but then we have to go through this process, next process of actual translation. Once again, if you need to take time, pause this so you can write this down, because this is very important. This is your major notes right here. So DNA to RNA to protein, and there's two processes. One is transcription, as one is translation. So let's give you an example of this occurring in our body. So take a moment, I'll pause this, and you guys pause this as well, so you can write down this example problem. All right, great, you're back. So we have this example problem. This is our original strand of DNA. So it's TAC, CGG, CAA, TCG. So this is super short. Now, when we do this, when we write it in 3-letter increments, these are called codons. So when you read it in 3 letters, it is called a codon. So if you're in the right corner or in the left-hand corner, a triplet is also known as a codon. So TAC is a codon. Now, the next part is to make the other side, because this DNA is double-stranded. So we got to make the next part, which is the complementary DNA, which is the other side of the DNA, to make the double helix. So our goal is, if you have TAC, CGT, CAA, and then TCG, you got to find the complementary. Now, when we talked about this a couple days ago, we always said that 2 things are going to happen. C is going to go with G. Those are always going to be the combination, and T is always going to go with A. So, which means if we do the first one, I'm going to do the first one and then have you guys do the next ones. T, the opposite of, oh, sorry about that. So the opposite of T is, you guessed it, A. Let me make this bigger, sorry. So the first one would be A. The opposite of A up here, the first one, is T. And the opposite of C is G. So your first complementary would be ATG, because it's the opposite of TAC. So pause this video and please finish the last 3 codons. If it's a C, put a G. If it's a G, put a C. If it's a T, put an A. If it's A, put a T. All right, let's see if you did this right. If you have a C, you should have a G. The G goes with a C, and T goes with an A. Excuse me. Following would be a G, a T, and another T. That would go with CAA. And finally, A goes with T, G goes with a C, and C goes with G. So fantastic. Our new complementary DNA strand reads ATG, GCA, G double T, AGC. That's our complementary DNA. So now we have a double helix on here. Now, this is part one. We're only down to part one, getting the DNA translated. Next up, in our next slide, as we saw right here, is we have to convert DNA into RNA. Now, you're like, oh man, this is super hard. What are we doing now? Well, it's actually really easy. As you notice, the only change between DNA and RNA is there a U. That's it. And it's only single-stranded. So this is like a single DNA strand. So when we go back to our example problem, this makes it really easy. So take your time and you're going to do your mRNA. Now your mRNA, this is called a messenger RNA, which is just a single strand of DNA. It's a very scientific word for a single strand of DNA. Except there's one exception. A's stay the same, G's stay the same, C's stay the same. The only thing that changes for mRNA is the U. T's are no longer used, it's U. So for this one, A stays the same, T does not stay the same, it goes to U. And then G goes to G, stays the same. So take this time, pause it, and for the next 3 sets of codons, if there's a T, place them with U's. Excellent, let's see how we did. So for the mRNA, the messenger RNA it's called, we have to see if there's any changes. Well, G's stay the same. C's stay the same. A's stay the same. So it should be the same. G's stay the same. T's we know do not. And the other T does not. So this should read GUU. And finally, A's stay the same. G's stay the same. C's stay the same. So between the complementary DNA and the mRNA, they're totally the same other than U's. So now we have this translation. Now the mRNA, aka Mr. Magniac in this situation, he's able to speak both languages. He's figured out what's going on. Now we've got to convert it into the complete other opposite. So for us, it would be like converting it into Spanish. Now we're not obviously speaking Spanish inside of our body, we're speaking protein. And this is where the new thing comes into factor. And this is where you have to go pick up this grid. It's called the DNA coding, and this is going to be a sheet on the front table. So if you did not pick it up, pause this and go grab. So welcome back. So we have our gene expression. You should have your sheet that's got all the letters in there. So let's do a back up here. We got DNA we started with. We converted that to RNA, which means we just changed our T's to U's. And then finally for translation, we got to turn everything into a protein. It's exactly how you guys think of like the protein you eat, exact same stuff. So how do we do that? Well, we do a little decoding. I feel like we're like doing a little mystery. I love mysteries. So we have to decode stuff. So let's look at our mRNA. That's our code that we have to decode. So we have AUG. This is 3 letters. This is coding for a different word, and the word is written in protein. So this is how we read it. So this is AUG. AUG, anytime we start this, we start in the middle. So we find the first letter. So over here we have 4 letters. We start with A because that's what it is. So we have A here. We'll do an A here. Next up, we find a U. So we go A, and then U. So go from the inside out. And then finally we find the third letter, G. We go A, U, or G. And this is our only option. And it is the protein or the amino acid called MET, which is methylene. Now we don't need to know that. All you need to know is how to use this code, this charting code. So this is like your translation dictionary if you're in Spanish class, or Google Translator. This takes words, in this case codes of mRNA, and translates them into another words in amino acids. So same thing as Google, except we're not doing it in English to Spanish. We're doing it from mRNA, which once again is a single strand of DNA, to protein, which is the protein that you guys eat, but you also make protein in your body. And that's what we're doing right now. We're making protein in our body. So let's try this next one out. G, gotta start in the center. There's our G. And we come back over here, we do C, C. And then finally A is the last letter. And no matter what, it doesn't matter. GCA is going to be, it's called ALA. So this is alanine, this is your second one. So I want you to stop this recording, and I want you to try the next two, GUU and AGC. So welcome back. I hope if you did your next set, which is right over here. Last time we did GCA. Now we're going to do GUU. So come over here. We have G, U, U. Ta-da-da-da. We have valine. So if we take and C, we have val. The last one, A, G, and then U, or AGC, I'm sorry, AGC, AGC. We have serenine. So this is how we speak in protein. This is what our body does all the time. Now, would this code do anything? No, you need 50 of these to make one single protein. Or the biggest one is, which you guys will watch a video, you'll see the biggest protein ever. So this is how we do it. So please finish the top of your sheet doing the gene expression. And when I get back to class, I can help you guys out as much as you need. But that is gene expression, the central dogma theory. Have a good day, and I will see you later.